Okay, so ladies and gentlemen, you are all welcome. So as we started the discussion that when we said the physiology is the study of how living organisms work, and then anatomy is about how the structure of cell, tissue, and organs look like. So when we say physiology is how living organisms work. So now the question is, what is the linkage between biochemistry and anatomy, biochemistry and physiology? So protein, protein is what determines the function of each and every living organism. Like for example, in human, let's take example with red blood cell. The red blood cell is responsible for the transport of oxygen. So what make the red blood cells to transport oxygen? The question is, or the answer is the hemoglobin. So hemoglobin is what make the red blood cells to transport oxygen. So you see, it means that the physiology of the red blood cell is the transport of oxygen. And what make the red blood cells to carry out that function is hemoglobin. And then the next thing, what is the anatomy? What is the anatomy? That is the structure of the red blood cell. It is biconcave in shape. So what make the red blood cells to have that biconcave shape is the hemoglobin. So that is why I wanted you to understand like physiology or biochemistry both in life sciences and medical sciences. Okay, thank you very much. I just saw you that you joined the YouTube. Okay, yeah. So try for those of you that didn't join the YouTube community, please try to join. So now the cell itself, or before you go to the cell, as I said, anything that is below cell, it cannot sustain life. So that is why virus is not considered as a living. It is just a particle. So it doesn't possess life. It only possesses some characteristics of life. That is why it is considered as a living. Sorry, that is why it considered some time to be a living inside a living host. Because virus, it can only replicate. It can only replicate. Okay, so Mia Chidima, thank you very much for joining the YouTube community as a member. So now, the question now is, can we kill a virus? No, we cannot kill what is not living. We can only deactivate it. So that is why supramolecular complexes, it is a level of organization of life that is below a cell. And that is why they are not considered as a living organism. And then more importantly, so ladies and gentlemen, in case if you have question, you can ask your question on the comment. You can ask your question on the comment section if you have a question. So always ask question on on your comment. So I will be watching them here. So now what I wanted to say. Yeah, Susan, I saw you. You are welcome. So now the cell itself. What is a cell? A cell is the structural and the functional unit of life. So anything that is living is because it has cell. And anything that doesn't have cell, it means that it is not living. And if you see something, or if you we as a human, if we are not feeling fine, or if you fall sick, is because there is a problem from our cell. If we are healthy, is because our cells are healthy. If we die, is because our cells are dead. So that is what I want us to understand. So anything is about how you able to understand how cells operate. And averagely, we have different types of cell, or we have uh, we classified organism based on their number of cells. So that is why we have multicellular organisms and unicellular organisms. Okay, Mary Daniel, you are asking how to join as a member. You can check the YouTube. You will see where you see something like join. So you click on that and you continue. So now the issue is I wanted to say that we have multicellular and multicellular. So when you said unicellular, it means there are those organisms that have only one cell. And when we said multicellular organism, they are organisms that have many cells. And we as a human, we are multicellular organism. Like for example, we as human, averagely, we have over 37.2 trillion cells. So these are the number of cells that we have in our body. But interestingly, what I wanted us to understand is that there are organisms that they have only one cell and they still survive because all their life processes are controlled by that single cell. So that is what I want us to understand here. So having one cell in some organism, it make them to undergo all the characteristics of living organism. But why am I seeing only 72 people? There are only 72 people that join the live. Okay, if you Isaac Blessing, if you want to join, you see joined on the channel and then you click on the join, so you'll become a member. So now that is about cell. And we also have two types of cell. We have prokaryotic cell and we have eukaryotic cell. So when you said eukaryotic, it means that they are cell that have nucleus and membrane bound organelles. So they have both nucleus and membrane bound organelles. So that is why they are called eukaryotic cell. When we said membrane bound organelles, it means that they have mitochondria, they have lysosomes, they have Golgi body, and so many others. And then we have prokaryotic organism. So when you said prokaryotic organism, it means that they are organism that doesn't have nucleus and membrane bound organelles. That is what you call prokaryotic organism. So okay, Jennifer Dima, you are welcome and thank you for joining the YouTube community. So now this is about the different types of cell. But I think, ladies and gentlemen, to make this of our lecture very easy, please just be asking questions on the comment section. I will be addressing them. I think it will be the best. Ask question because this seems to take us long. Ask question. Yes, Obioma Esther. Protein determine the function of every organism. Like for example, let me just give this example. We as a human, we can talk. Why do we talk? Is because we have a protein that are actually expressed in our body. That is why we as a human, we talk. There is a gene called FOXP2. So we as a human, we have a gene FOXP2 that produce a FOXP2 protein that make us to talk. And there are organisms that they cannot talk because they doesn't have that. And in addition, we as a human, we can think, we are intellectual, and at the same time we have a cognitive domain. Why? Is because they are protein and the neurotransmitters in our brain. That is what we think, we read, we interpret, and we understand things. Is because of the protein that are present in our brain, or we can say neurotransmitter. So Elizabeth is asking question about protein purification. So I think Obioma Esther, you are now able to understand that protein is what determine our body function. So then now the next thing is uh Olayemi, you didn't join the YouTube community. So you asked about protein purification. So what is protein purification? So generally, most of the proteins that are in our body, they are inside cell. Some they are even inside organelles of the cell like mitochondria. So assuming now you want to get a protein that is in the mitochondria and you want to get it outside that cell, you want to purify it. Let's say, okay, let me just give this example. Which protein? We know that we have insulin injection. We have insulin injection. But now the question is, okay, Fumilayo, you are welcome and thank you for joining to the YouTube community. Good evening. The question is, how do we get insulin from human? Although the insulin that we are using in the chemist for treatment of diabetes is not actually is a recombinant insulin, is a recombinant insulin. But now assuming now you want to get insulin from me, from I as Mukhtar, you want to get insulin from me. How do we get it? You need to follow this protein purification procedure. So how do you do it? Number one, which organ do we produce pancreas? Do we produce insulin? Please, who can answer this question? I want to see the answer on the comment section. Which organ produce insulin? Which organ produce insulin? Someone should answer it. Excellent, Chiamaka, pancreas. But specifically, how, which of the cell, of the pancreas produce insulin? Is it alpha cell or beta cell? Excellent, is beta cells. Is beta cells. So now, as Mukhtar, and you want to get insulin from me, how do you do it? First, you need to get my pancreas. So after you get my pancreas, what do? You need to homogenize the pancreas. After you homogenize the pancreas as a tissue, as an organ, you homogenize it to the tissue, and then you later also further get the cell. So in the pancreas, we have so many cells. We have alpha cell, we have beta cells, we have gamma, we have epsilon cells. So which cells do we get insulin? Is the beta cells. So you need to get the beta cells from the pancreatic tissue. So after you now isolate the pancreas, after you isolate the beta cell, what do you do next? Who can tell me? You now have the beta cells, and what you are after is to get insulin. So what do you do with the beta cells? For those of you that watch the video, what do you do? You get, after you have the beta cells. So how do you get insulin from the beta cells? You see, you homogenize, and then you have the beta cell. After you have the beta cells, I need answer. What do you do with the beta cell? I'm waiting for an answer. You have the beta cells. How do you get the insulin from the beta cell? Okay, someone said no idea. Someone said homogenize it. Yeah, you need to get what we call a suitable isolation media. When you said a suitable isolation media, of course you need to go to further homogenize. You need to open up the beta cell, but you need to check from the literature in the beta cells, where does it specifically located? Is the insulin produced in the cytoplasm? Is it in the mitochondria? Is it in the Golgi body? If it is in the mitochondria, you need to open up the beta and then you centrifuge and then you get the mitochondria. But generally, insulin, we get it in the cytoplasm. So after you open up the cell, you need to centrifuge and get your supernatant as your cytoplasm, and that is where your insulin will. And that is not where, of course, in the cytoplasm, there are so many other proteins. There are so many other proteins. But what you are after is to isolate and purify insulin. So, what do you do to remove, or how do you do it to just get insulin and discard the remaining protein? So in the cytoplasm that you get after homogenization and centrifugation, there are other proteins, there are protein component, and there are even non-protein components. So what do you do? The next thing is precipitation. You need to precipitate the protein. And how do you precipitate the protein? Is by adding a salt. How do you add the salt, or which salt do you use? You use ammonium salt. So when you use ammonium salt, it will precipitate all the protein in the cytoplasm, and then the non-protein component will not be precipitated. So after you do that, then you do what you call salting out. That is dialysis. So you take everything and you put it in the dialysis bag. So all the non-protein component will be removed and you will be left with the protein. So the protein will be precipitated. So the next thing, after you have your precipitated protein, then you follow it up with other purification techniques. So what are the purification techniques in protein purification? We have column chromatography. We have size exclusion chromatography. We have affinity chromatography. These are the different types of purification techniques, and each of these chromatographic techniques have its own principle. So you need to have an idea of what your protein is. You need to understand the physicochemical properties of your protein. So you can, from there, you can decide to say, okay, you'll use affinity chromatography. So when you use affinity chromatography, it means that you will take all those precipitated protein and you will now put it inside, or you now place it into affinity chromatography column. So all the proteins that are not actually, because in the affinity chromatography you have like a ligand inside the chromatographic techniques, and the ligand is uh it can be compound that your protein of interest can be bind, and all those proteins that are not of interest, that is those proteins that cannot bind to the ligand, they will be eluted. That is, they will pass, and then you can get your protein in a pure form using affinity chromatography. Using affinity chromatography. Or you can use size exclusion chromatography. That is based on the size, based on the size of your protein. Okay, so San Ogbo, the purification techniques, it started from dialysis. Okay, it started from one, homogenization. Homogenization, number one. Second, centrifugation, then precipitation by ammonium, affinity chromatography, size exclusion chromatography, column chromatography. Apart from this, then there is also what we call uh, so electrophoresis. That is electrophoresis. So these are the protein purification techniques of Bo. These are the protein purification techniques. So now after this, then you can decide to say you will use affinity chromatography, or you can decide to say uh, it's not colon, it's column. Olayemi is column chromatography, not colon. Column chromatography, COLUMN, that is column chromatography. That is the protein, yes, that is the protein purification technique. So each of these protein purification techniques have their own principle. Affinity chromatography is based on the binding. There is also ion exchange chromatography. I didn't mention it. That one is based on the charge of the protein. There are proteins that they are negatively charged, and there are proteins that are positively charged. So if your proteins are negatively charged, so in the ion exchange chromatography, you need to have a bead or a column that is positive. So the negative proteins will bind to the positive uh, exchanger, positive exchanger. And if the protein is negative, then you need to have cationic exchanger. A cationic exchanger is an exchanger that have positive charge. So only negative protein can bind to it. And then for the positive charge protein, then it is cationic exchanger that you will use. That is the positive charge cation exchanger. And then for the size exclusion is based on the size of the protein. And then for the column chromatography is based on the mobility, the movement of the molecules. That is column chromatography. So you see, this is the principle that you need to understand for each of the techniques. And then for the electrophoresis is based on molecular weight. Sorry, it's based on ion. Sorry, it's based on charges too. It's based on the charge of the protein. So this is actually a brief introduction of for you that actually ask question about a brief concept of protein purification. This is it, and I send the video on that. You can go back and watch that video for you to have more understanding. This is just a brief introduction. So who have any other question? I addressed this. So what other question do you have? I'm listening, right? If you have any other question. For those of you that didn't join the YouTube channel, try to join. Try to join the YouTube channel. Okay, Bola Tosin is asking I should help to classify amino acid. Okay, so I will do that. So amino acids, they are the building block of protein, and they are classified on different bases. The number one, we have essential amino acid and we have non-essential amino acids. That is based on the nutritional requirements. There are amino acids that we cannot synthesize them by our body. That is essential amino acids. We cannot synthesize them in our body. We get them from diet. And we have non-essential amino acid. They are amino acid that we can synthesize them in our body. And we also classify amino acid based on how we utilize them in our body. So we have what we call glucogenic amino acid. They are amino acid that can be used to synthesize glucose in our body. That is what we call glucogenic amino acid. And we have, apart from glucogenic amino acid, then we have, uh, okay, please, sir, can you direct me on how to join the YouTube channel? I have been trying to join, but it is not working. Thank you, sir. Yeah, when you check the YouTube channel, apart from subscription, you will see join. So you click on that and you join. You follow the procedure and then join. So what I'm saying now is that I'm saying okay. So we have glucogenic amino acids. So they are amino acid that you can use them to synthesize glucose. And we have what you call ketogenic amino acids. They are amino acids that can be used to synthesize ketone bodies. So what are ketone bodies? Ketone bodies, they are molecules that apart from the glucose, the brain can utilize it to produce energy. Like brain, apart from the glucose, it doesn't use any other class of macromolecules to generate energy. But sometimes it can also utilize the ketone bodies. So glucose and ketone bodies are the major biofuel that are utilized by the brain to generate energy. And then we have amino acid that are both ketogenic and glucogenic. That is the amino acid that can synthesize glucose and ketone bodies at the same time. And we also classify amino acid based on their chemical structure. So some amino acids, they are classified based on their structure. So that is why we have non-polar amino acids like glycine, alanine, isoleucine. And we have polar amino acids like serine, cysteine. We have basic amino acids. We have lysine, we have asparagine, and we have acidic amino acid, aspartate. Isomer is not glycogenic, it's glucogenic. It's glucogenic, not glycogenic, it's glucogenic. So apart from that, so these are actually the major classification of amino acids. So who have another question again? So this is the classification of amino acid briefly. So who have another question again? Okay. Olayemi is asking question, what transport system is the release of insulin in pancreatic cells? Yeah, you know that we generally synthesize insulin from the pancreas. So after we synthesize insulin from pancreas, so how did we release that insulin from the pancreas to the bloodstream and transport it to the target cells? Yeah, of course, we have talked about the different types of transport system. We have passive transport, we have active transport, we have facilitated diffusion, we have simple diffusion, we have also osmosis, and then we have uh, active transport is divided into primary and secondary, and we have bulk transport which comprise of endocytosis and exocytosis. So now the question is, how do we transport protein? So is protein a non-polar or non-polar? So you need to understand that. So most of the proteins they are polar, so therefore they use either a carrier transport, so mostly actually is active transport or pacilitated diffusion. So active transport or pacilitated diffusion are the types of they are the types of transport that require or that usually transports a polar molecules, water soluble molecules. So it's usually active transport or pacilitated diffusion. So that is the answer to this. Susan Ogbo is asking, are you saying there are five classification of amino acids? The classification of amino acid are more than, sorry, I actually gave the classification on three different bases based on their polarity, and then also based on their nutritional requirement, and then based on their body demand. Expensive Diago, thank you very much for joining my YouTube channel as a member. Thank you. So, what other question? Okay, sir, please explain more on the assignment on LMS 50 word for each of six topics summary. Yeah, what I want you to do is try to for each of the video summarize it in 50 words. So a total of six topics, it means that you are going to have 300 words in all. Isoma is asking the difference between active transport and facilitated diffusion. Active transport generally require energy for them to transport molecule. They require the use of energy, while facilitated diffusion require a channel or a carrier. But for the facilitated diffusion, it require only a protein carrier or a channel for it to transport a molecule. So you see, ladies and gentlemen, try to be understanding this. Active transport require ATP, that is energy, while facilitated diffusion doesn't require energy. But in terms of carrier or a channel, both of them use a carrier or a protein channel for them to transport a molecule. Okay, Maimunat Muhammad, thank you very much for joining the YouTube community. Please, sir, you asked questions earlier. Does physiology of sleep love part of our course outline? Because I didn't see them in our course materials I have. Yes, physiology of sleeping and biochemistry of love are not part of the course material. I did that for international for the students that are international, and for also some of you that are interested with the lecture. So it's not part of your lecture, and nobody would ask you that in an exam or test. So it's not part of our lecture. That one is for general knowledge for those of you that are interested. Okay, Chinwendu Wahiri, you are welcome. Okay, just give me 1 minute. I'm coming. So, okay, someone said, sir, explain more about isomers. So, when we said isomers, it means that generally they are different. So, that is what we call isomerism. So, most of the isomerism that is related to the biochemistry is stereoisomers and stereoisomerism and geometric isomerism. So, when you said stereoisomerism are compounds, the first they must have or they must fulfill some criteria because before we consider them to undergo optical isomerism or stereoisomerism. Number one, they must have chiral carbon. So what is a chiral carbon? It's a carbon that have four different groups attached to it. Wow, Chinyere Agu, thank you very much and welcome for joining my YouTube community. You are welcome. Thank you. So that is the concept of, I forget my what am I saying? Okay, they must have four different groups attached to them before they are considered to undergo stereoisomerism. And I think I gave you formula on how to calculate stereoisomerism. Like in glucose, there are, I think, there are four chiral carbon. So how did you calculate the number of stereoisomerism? Is 2 raised to the power of n. So n indicates the number of chiral carbon. So if you have 2 power 4, that will give us 16. So it means that there are 16 stereoisomers of glucose. And of course, also at the same time, these stereoisomers, it can be L isomer and it can be D isomer. So you can go and watch video on that because I have made a video on that. Unless if you have more question, but of course, in the case of um, geometric isomerism, they have a restricted movement because they have a double bond between the two carbon, so they are restricted, they cannot rotate. And it's also depend on the direction of the group. So we have trans isomerism and also cis isomers, trans and cis geometric isomerism. So you can go and watch the video on that briefly so that you actually get it. So Tosin said, I don't seems to have the major classification of amino acid in my notes, sir. Okay, so if you don't have it, I will advise you to go and watch my video on the concept of amino acids. I have video on that, and I think I send you so you can go there and maybe you can even talk to me privately. Or maybe I will just send it to the group after this class. Okay, Erugo said, I want to join to the YouTube channel. How do I go about it? Yeah, when you check the YouTube channel, you will see where you are going to subscribe. There is a place where you are going to join. So click on that and then continue. Okay, Diago, expensive Diago, please, sir, can you explain more on nucleic acid, nucleotide? Also, I don't understand the structure of purines and pyrimidines. So when we said nucleic acid, they are polymer of nucleotide. So nucleotide, or let me just start with this. Nucleosides is when you have sugar and nitrogenous base. So nucleosides is when you have ribose sugar plus nitrogenous base, you have nucleosides. Then when you have nitrogenous base, phosphate sugar, and sorry, when you have nitrogenous base, ribose sugar, and phosphate group, when you have these three together, then you have nucleotides. So it means nucleoside plus phosphate is what gives us nucleotides. And when nucleotide, okay, someone said I should come again, Hassan Shafa. Okay, I said nucleosides. Nucleosides is when you have nitrogenous base and pentose sugar. You have nucleosides. But when you have pentose sugar, nitrogenous base, and phosphate group making three, this is what you call nucleotides. Nucleotides. So it means that if you have nucleoside plus phosphate sugar, that will give us nucleotides. And then what are these nucleotides? The nucleotides, they are the building blocks. They are like blocks that you will use to construct nucleic acid. So it means that a nucleic acid is like a building that is constructed with nucleotides. And these nucleic acids, we have DNA and RNA. DNA is deoxyribonucleotides. Then we have RNA, which is ribonucleotide. So what is the difference between DNA and RNA? The pentose sugar in DNA is deoxyribose sugar, and then the pentose sugar in RNA is ribose sugar. So you can still go back to my video and watch the concept of the nucleic acid. You will get more clarification because there are structures there that can actually, or that can easily give you a full picture of what they are or what they mean. So what is there any other question? But in case of how much you are welcome. We didn't join the YouTube community. Sadiq Omar, good evening. Yes, good evening. How are you? Any other question? Yeah, I said that I will be doing the revision by asking questions. So if you have any problem with any topic, then you can ask. Yeah, she's also Diago is asking about the structure of purines and pyrimidines. So that is why I say you should go back to my lecture. There are a video there. There are video that show the structure of purines and pyrimidines, and you can easily understand them. They are heterocyclic compound, and these purines, we have two pyrimidines. Sorry, we have three pyrimidines and we have two purines. Purines are adenine and guanine, while pyrimidines we have thymine, cytosine, and uracil. Uracil is found in only RNA. So uracil and cytosine, they are pyrimidines that are found in RNA, while thymine and cytosine, they are found in DNA. And generally, these nitrogenous bases or the nucleotide, they pair together. They pair together to form, or yeah, they actually go what we call a base pairing based on Chagap's rule, where guanine pair with cytosine using a 3 hydrogen bond, and then guanine and cytosine 3 hydrogen bond, adenine and thymine through 2 hydrogen bond. That is the base pairing. Okay, thank you, sir. Please, can you help explain the lecture on carbohydrate? It is a bit complex. Wow. So you see, we said carbohydrate is a polyhydroxyaldehyde or polyhydroxy ketone. They must have either aldehyde functional group or ketone functional group. And of course, they must have fully hydroxide. There must be so many OH group in them. Yeah. So we also have different classes of carbohydrates. We have monosaccharides, we have oligosaccharides, and then we have polysaccharides. So I always advise some of the lectures that you should watch them. Because I just wanted you to ask directly where you have problem. Because now if you say that I should explain carbohydrate, it means like I have to repeat the lecture again. Unless if you have a specific point where you have problem. Adamu Shafa, you asked about stereoisomers and optical isomers. Stereoisomers is one of the type of optical isomer. So it's a subtype of optical isomer. Okay. Diago is asking uh, a nucleotide containing one or three phosphate. We have adenine monophosphate, or we can have guanine monophosphate. That is guanine containing only one phosphate. And we have ATP, that is adenosine triphosphate. That is the example of nucleotides that have three phosphate. We can also have GTP. GTP, that is guanine triphosphate. We can also have GDP, guanine diphosphate. That is when two phosphates are there. Okay, so what protein have to do with the etiology of edema? Wow, edema is a kidney disease. If there is colossal accumulation of the kidney, of course it will inflame. There will be inflammation. Okay, expensive Diago, yes, thank you too. So any other question? Any other question? Okay, Elizabeth Olayemi, where did you see synthetic nucleotides? Isaac Elizabeth, I have explained about the amino acid. I explained to them. Thank God that you even asked me about this amino acid. I think I have some video that you have to watch on amino acid, which I didn't actually, I forgot to send it to you. Yeah. Okay, I should explain it again. Don't worry, I have one video on amino acid. After this class, I will definitely send you. I will send you. Okay, so synthetic nucleotides, they are nucleotides that are synthesized in the lab. We cannot actually, or our body cannot synthesize them naturally. Rather, we synthesize them in the lab for a specific purpose. Yeah. Yeah, Precious Smart, I will send you a video on amino acids. I think I have to send you like 3 different videos on that amino acids. Yeah, I will send you those videos. So any other question? Okay, Smart. Precious Smart, don't worry, I will send it. Okay, why did you see duplication and RNA in nucleic acids? Yeah, histidine is an essential amino acid. You cannot synthesize it in our body. Because when you say duplication, is it gene duplication? Is it what you mean? But Diago, yes, I was studying the course material and I saw it. Yeah, please send me that course material, Diago. Yeah, I want to see it. So please explain human genome. Yeah, when you said human genome, it means the complete genetic makeup of a living organ, which comprises the complete DNA of human. Like, for example, the human genome is different from the other species genome, and that is why we are unique, or we are different from other organisms. It's like it is the signature of the species. So each of a living organisms exists at the species as it is because of this genome signature. And with the genome, if you can get the human genome, you can easily construct a complete human being. And of course, in the human genome, it comprises about 3 billion base pairs. That is the nucleotide. There are about uh, 3 billion base pairs. 3 times 10 raised to the power of 9 billion base pair. Lisa, what process dot red blood cells being transported? RBC transported in the cell. No, Okudo, this your question is not correct. Red blood cells is a cell, so how, what do you mean that how is it transported in a cell? It is also cell on its own. So I think we have tried, Aiko. We can stop here. Maybe you can still ask the question, or maybe I will be asking you in the group. So for those of you that didn't actually join the YouTube community, please try to do that. If you didn't join the YouTube community, please try to do that. So can we end the live now? If there is no any question, can we end it? Do we need to still go back to Google Meet? Balkisa Hamasi is saying, Sir, this your knowledge, how I wish I am the one. Wallahi, read every day. If you can read every day, that is the key to success in whatever you are going to learn. So read every day. Try to develop a habit of reading every day. So if you actually develop of reading every day, you will learn a lot of things. Today I was on TikTok and I was shocked when I saw someone make a particular comment. Although he blocked me, I don't know. I don't need boss. I can't accommodate all of us. Okay, it can't accommodate all of us. Yeah. So what I want to